featuring Dr. Sheldon Cooper. Mm-hmm.
Okay, so now I'll tell you about how securin and separase promotes progression through the cell cycle to the point of sister chromatid separation. As we can see here, two sister chromatids, not at the centrosomes, are tied together through a protein that will have to be degraded in order for them to separate. The pathway for doing this is a cooperativity between separase, securin, and other proteins or enzymes in the cell at this time. So we know from previous reading that APC has a property of ubiquitin ligase activity. Under specific conditions, APC is bound to a substrate specificity factor, CDC20, that interacts with inactive separase, as shown here and represented through a junkyard bulldog with a muzzle representing securin. So under APC activity, when bound to CDC20, it adds ubiquitin ligase molecules represented here in blue to securin, alleviating and taking off the muzzle on separase, activating its activity, shown here through the angry bulldog. So the effect of this activity is active securin can literally bite through its substrate that is tying the two sister chromatids together. When it does this, the two sister chromatids go their separate ways. However, at the centrosomes, you don't want to do this just yet, so an additional adapter molecule called PP2A, protein phosphatase 2A, acts effectively as a chain link fence, blocking even active separase from biting through the protein that is still having the two sister chromatids bound together at the centrosomes. Additional mechanisms need to be active in order to dissipate this protein phosphatase 2A activity in order for separase in its active form to effectively bite through the tethering protein at the centrosomes, which is progressed later at a later point in the cell cycle. The end.